Thank you very much for that introduction, and I really am humbled to be here, to be honored by the Leopoldina and to give a talk in front of this audience, which happens to also include kind of the founder of the field that I work in, so I'm doubly nervous to give the talk in this specific context, but we'll come back to that. So, I only have 20 minutes, and I've been warned to speak slowly for our translators. So let's get right into what we work on, which is the evolution of protein complexes. Oh, micro. There we go. I'm sorry. So to start us off, let's look at this drawing of an E. coli cell, a common gut bacterium in humans, from David Gutzel. And if you look at the zoom of its inside, you can see that it's teeming with these complicated-looking shapes. And if we look into these are protein complexes. So those are macromolecular structures that are made of several polypeptide chains that associate together and then make one functional unit. And the top one that you see there is an ATPase that takes an ion gradient and turns it into the cellular energy currency ATP. And the bottom one is a metabolic enzyme. And to evolutionary biologists like me, these structures, even though they're small, are kind of a representation of the more general phenomenon of biological complexity. If you think about it, each of these polypeptide chains has to fall into a very precise three-dimensional shape that involves hundreds of precise contacts, and then these chains have to find each other by self-assembly in this total chaos that happens in the cell to make this very specific functional unit. And what we're interested in is how and why evolution generates these structures. And there are broadly three reasons for why we find this interesting. One of them is that we wonder whether these structures can only emerge in evolution if they have some kind of functional use that natural selection can work on. Or whether perhaps sometimes structures like these can come into existence and then also persist without any use at all, just by chance. And we'll hear a little bit about that later. A second reason to study the evolution of these structures is that it can teach us about the relationship between their amino acid sequence, their structure, and their function, which is something that genuinely interests biochemists. And in the abstract, that question is still very hard to answer, how a specific function emerges from any specific sequence, because these objects are chemically and physically very complicated. But a much simpler question we can answer today is how an evolutionary change in a sequence leads to a change in a structure, which then gives you a new function. And then the third reason that brings us to studying the evolution of these entities is that the emergence of some protein complexes has perhaps changed the history of life more than any other or most other evolutionary innovations. And we're just interested from a historical perspective exactly how that happened. But of course, there's an obvious problem with this. How can we know anything about the history of molecules? These entities are too small and fragile to leave any fossils, so we have no direct access to their histories. Fortunately, information about their histories is still encoded in the sequences of proteins that are alive today. And we can retrieve it in a process called ancestral sequence reconstruction, which Stephen Benner, who's I think in the audience, pioneered. And here's how this works. We first generate what's called a multiple sequence alignment, where we line up the sequence of the same protein from many different organisms, from which we can then infer a phylogenetic tree. And this tree is a family tree of these proteins that describes their genealogical relationships, and where the individual lineages of different proteins meet at these internal nodes, those correspond to ancestral proteins that would have existed in some now long extinct organism that lived hundreds of millions or billions of years ago. And it turns out that we can use the alignment, the tree, and a probabilistic model of sequence evolution to estimate the sequences at these internal nodes. Once we know the protein sequence that would have existed in the past, we can ask a company to synthesize a gene that codes for it, give that to an organism that can turn it into a protein, isolate that protein, and then we can interrogate its biochemical properties in the lab to learn about the ancient biochemical past. And in this case, this is something that I was involved in as a postdoc. We made a very ancient version of hemoglobin, the blood pigment that carries oxygen through our blood, to understand how that originated. Okay, that's the technique, and I want to give two examples of how we use this to understand protein complex evolution. And the first one has to do with rubisco. This is an enzyme that helps plants, algae, and cyanobacteria fix CO2 and make sugar out of it, which ultimately almost all of life depends on. Virtually all of the organic carbon that you see around you in this room or outside in nature has at some point passed through the active site of this one enzyme in the form of CO2. And that means that even very small changes in exactly how it catalyzes this reaction can have history-changing consequences for life on our planet. And the theme of this enzyme's history is that it doesn't just react with CO2, it has a problematic side activity with oxygen. And this happens because to this enzyme, oxygen looks a little bit like its substrate CO2. And when it reacts with this gas, it makes a very toxic side product called phosphoglycolate. And the problem with this chemical, to simplify things a lot, is that it is an inhibitor of central carbon metabolism. And if you accumulate even small amounts of this, you can totally shut down photosynthesis. Rubisco has this activity because it's much older than photosynthesis. And it first emerged in an atmosphere that had no oxygen at all, where it didn't need to distinguish between these gases. But in order for it to become part of photosynthesis, which of course produces oxygen, it had to learn how to get better at distinguishing between these two gases. And we can still see the outcome of this historical adaptation process in the properties of rubiscos that we find today, in their so-called specificity constants. And this number quantifies the relative preference for CO2 over oxygen. The larger the number, the higher the preference for CO2. And in this graph, at the bottom, you see the specificity constants of more ancestral type anaerobic rubiscos that never learned how to cope with oxygen. And at the top are all the oxygen-adapted rubiscos, and there's a big gap in the middle. And for a long time, it wasn't clear how evolution crossed that gap. It's obvious that it's important because you have to cross that gap so as to not poison yourself with oxygen. But these enzymes have identical-looking active sites. Where the reaction occurs, all the amino acids look the same. So it wasn't clear how evolution managed to cross that gap. That's also a technologically relevant problem because rubisco continues to react with oxygen even today, and that's a property we would like to get rid of in the enzyme. So if we could learn how evolution manages to make it more specific, perhaps we could use that knowledge to eventually make better enzymes in plants and increase our agricultural yields. So for a long time, people didn't know what distinguished these two classes of enzymes, and there was only one hint, which is that these kinds of enzymes make different complexes. The more ancestral type rubiscos look like this. They are formed of a basic unit that's a dimer, so that's two polypeptides coming together. That's in red and green here, and then several dimers can be arranged in a kind of barrel, and that then catalyzes CO2 fixation. Whereas all the oxygen-adapted ones have an additional protein component, the so-called small subunit. They are also made of dimers of this catalytic subunit that are arranged in a barrel. So it's 4 dimers for a total of 8 subunits. And then that's capped at the top and the bottom with this new subunit. And this doesn't bind anywhere near where the reaction actually occurs. It's not near the active center, and this new subunit isn't chemically active itself. But it's the only obvious structural difference between these proteins, so people long suspected it had something to do with increasing the specificity for CO2. And this theory was untestable for a reason that's both common in biochemistry, annoying to biochemists, but also very interesting from an evolutionary perspective. What you would want to do is to take one of these specific rubiscos and ask how specific they are if you take away that new subunit. But that's not possible, because if you try that experiment and you try to make this protein without its new friend, you get a completely insoluble and an active enzyme. In a sense, these rubiscos have become completely reliant on this novel component. They've forgotten how to exist without a component that previously wasn't needed. And that gets in the way of our causal inferences. It's also interesting to think about why this happens at all, why proteins forget how to exist without novel components, but that's perhaps a question for another day. So we thought we could tackle this problem by using history to work out what happened when this new component was added. And here's how that worked. So this is now a family tree of the rubiscos, and in these phylogenetic trees, time runs from the root, which is on the left-hand side of the tree, all the way at the back, into its leaves, so from left to right. And we could see that on the bottom here, we have a family of rubiscos that always encode its catalytic subunit right next to this additional component. There are these large and small arrows. And next to them are their distant cousins that lack this subunit. So we could time the acquisition of this novel component to the phylogenetic interval that's highlighted here in orange. And that allowed us to infer the sequence of 2 key ancestral proteins. One of them I'm going to call Ankh-LS for the ancestor of a Rubisco that had a large and a small subunit. And the other one I'm going to call Ankh-L, which is its immediate predecessor that was lacking this component. We then produced these proteins that are more than 2.5 billion years old in the lab and characterized their structures. And this is a cartoon of what their structures look like. The younger of the two ancestors, Ankh-LS, looked like a normal, highly specific Rubisco with that novel component in it, whereas its predecessor was lacking that component, but still made a barrel-type complex. We then measured their specificities, and we found that the younger of the two, Ankh-LS, already had a very high specificity, similar to that of a cyanobacterial Rubisco that would be part of photosynthesis, whereas the older of the two, Ankh-L, was still unspecific. And that meant that we have this temporal correlation here between recruiting that novel component and becoming highly specific. The question is if that connection is causal or if it's just a coincidence that both of them happened at the same time. So what we needed here was a protein that could bind the novel component but wasn't totally addicted to it yet. And neither of these two proteins fulfilled that criterion. So the green one couldn't bind that new component yet. It hadn't learned how to do it. And the gray one was already totally addicted. So what to do? These two proteins are separated by a set of 95 historical substitutions. And that's just the differences in their sequence, the number of differences in their amino acid sequences. And among those 95 differences must be some substitutions that allow this new interaction, and potentially some others that create this addiction. And the hope was that we could separate those two sets such that we get a protein that can bind the small subunit but doesn't need it. In the end, we managed to do this after a lot of trying, and we found 7 historical mutations that we could put into that protein that allowed this protein now to interact with the small subunit without needing it. And that's what this graph here shows. This is just measuring the specificity of this protein in the absence and the presence of the new subunit. That's the minus and the plus. And you can see that giving it this new protein doesn't lead to a big increase in its CO2 specificity. So something was missing, and just binding this new protein was not enough. And we found what was missing, and it was very surprising, kind of by accident, which was that we were just missing one more historical mutation, which had a very bizarre effect. If you make that mutation in the absence of this new component, nothing happens. But it does increase the specificity massively when that novel subunit is bound. And this mutation is nowhere near the active site of this enzyme. So what does this mean? In genetics, we call this phenomenon epistasis, which refers to the context dependence of mutations, who can have some effect that's drastically different depending on what sequence context you make the mutation in. In this case, this novel subunit allosterically modulates the effect of this other mutation. That's why we call this an epistatic modulator, and that's really its effect on the evolution of Rubisco specificity. And we're now hoping to turn this insight into new protein engineering approaches that can help us increase the specificity of Rubisco further. So just to summarize this quickly, what we learned from this kind of approach is that Rubisco cleared this key hurdle in becoming part of oxygenic photosynthesis a long time ago. Its novel subunit enabled this, but not directly, but in this weird indirect way, in that it modulated the effect of mutations that happened afterwards. And we could only find this out because we overcame this essentiality problem by going back in time and working out what effect components have when they were first added. Okay, so for the remainder of my time, I'm going to switch gears a little bit and talk about something quite different. Which is a question more about why things are the way they are today, more than it is necessarily a question about history. And this is about the phenomenon that many enzymes form so-called homooligomers. These are protein complexes where you take the same polypeptide chain and you arrange multiple copies of it into a complex. Many enzymes do this, and sometimes there's a really good reason for it. For example, the active center could be at the interaction site between two subunits. Or sometimes this kind of association allows you to regulate the enzyme allosterically in a way that you couldn't do that if you just had a single chain. But there are very many enzymes that have one active center per subunit and no known allosteric regulators that depend on this association into a complex. And that poses the question, why do this? These are complicated-looking structures. Is there another reason? And for a long time, people have suspected that perhaps these structures can just arise by chance, as long as they're harmless. And we're going to come back to that idea of whether it's plausible that something so complicated could arise by chance in a second. But this motivated us to ask how strong this linkage is between what kind of complex an enzyme makes and its function. And we decided to work on what we thought was a very boring enzyme called citrate synthase, which is part of the Krebs cycle that you use to break down nutrients. And we chose this enzyme because it always does the same job in nearly all organisms. And it just interested us, how much does it vary, what kind of complex this enzyme makes. And we made very many bizarre discoveries in this enzyme family, which I will not have time to talk about. I'm just going to talk about the very strangest thing that we discovered. And this was one particular citrate synthase that we took an image of with an electron microscope. And what you see here is one of those images, or many images averaged together. And every white blob is actually 2 protein chains, making this strange triangle here. So that's 18 proteins making this weird triangle. And in the beginning, when we took this image, I didn't know what I was looking at. I just thought it looked funny because it's a triangle. So I sent this to a physicist friend of mine and said, look at this weird triangle that I found. And he looked at it and he said, hang on, that looks like it's a triangle of triangles. Because to him, the corner of this triangle, which is made from 6 subunits, looks like a smaller triangle that you then arrange into a bigger triangle. And that reminded him of a famous fractal pattern. That's a mathematical pattern. We'll come back to that in a second, called the Sierpinski triangle, which is an infinite series of triangles. So he asked us, on your electron micrographs, do you see any larger triangles that would be 3 times the thing in the middle to make an even bigger one? And of course, eventually we did find them, and we found these huge molecular complexes. This is made of 54 proteins that associate together with this giant void in the middle. We were eventually also able to solve atomic structures for these, or near atomic structures for these triangles. But at that point, we had convinced ourselves that we had found a molecule that can self-associate into a Sierpinski triangle. Now, why is that significant? The Sierpinski triangle is a fractal. Fractals are mathematical structures whose salient feature is that the level of detail you see does not depend on how far you zoom in or out of the structure. You always see the same amount of detail. These structures never become smooth. And there are many fractals in nature. Most of them are of this kind, like riverbeds or drying paint, the cracks in drying paint, which are irregular. In these structures, what you get is a similar branching ratio from a large channel in a river to a smaller channel branching into even smaller channels. But the correspondence at different structural levels is not exact. Same for the cracks in drying paint. Regular fractals, in which the large structure is an exact copy of the smaller structures that it's made of, are incredibly rare in nature. And I guess the most famous version of this is the Romanesco broccoli, which you may have seen images of on the right. And there had never been any observation of a regular fractal at the molecular scale. In fact, there were theories about that this would be mathematically impossible because you can't make these fractals using algorithms that map well on how molecules self-associate. So we found ourselves in a bit of a paradox that we had apparently just stumbled on the first regular molecular fractal. And what I want to spend the last 3 minutes of my talk about is whether this is complicated. It's clearly rare, but is this difficult for evolution to build, and do you need a function to build it? Clearly, your eye is such a complicated structure that it wouldn't spontaneously arise in evolution unless it had a use. You need thousands of genes to act in concert to make your eye during development. So that requires the deterministic power of natural selection to build that structure. But perhaps, as beautiful as these structures look, maybe these are easy enough to construct that they could emerge by chance. So we tackled this again by just studying how this structure evolved using the same approach as before. So we built a phylogenetic tree of the protein that made this fractal and all of its relatives. The protein that makes the fractal is this blue little triangle on the tree. And again, time runs from the root of the tree up at the top into the leaves. So what we decided to do is to work our way from the protein we started with into the past and just ask how early on did these proteins learn how to make a fractal. So we resurrected some ancestral citrate synthases and characterized their structures. And these first 2 nodes, the blue and the green one, they could still form these fractals. And everything before them could only make a very small complex and was unable to make these fractal structures. And that told us that it was between these two ancestors that the protein learned how to make a fractal. And looking into the biochemistry of these structures, we could then work out that all it took was a single historical mutation to turn this boring structure into something that can make a fractal. So genetically, as simple as you could imagine. We then did a whole bunch of genetic experiments on the bacteria that harbor these complexes to ask whether they care about if this protein can assemble into a fractal. And the answer was no. We could replace this protein with something that could not make fractals, and the bacteria grew just as well. Now, that's not hard proof that there isn't some situation in which you might need assembly into fractals, but it's certainly suggestive. The other thing we discovered is that this fractal assembly was very quickly lost in evolution in multiple lineages, so it only lasted in a single bacterium after its evolution, which in a sense made it even more remarkable that we just stumbled on it by accident. So to us, this specific example of a complex evolving has all the trappings of a structure that might just have come into existence because it did no harm. It was so easy to create in a single mutation that it could fix through what in evolution is called genetic drift. And genetic drift can sometimes let a mutation take over the population, even if it doesn't do anything useful for the organism that it evolved in. So that, in a sense, changed our perspective on whether we should always attribute function to complicated-looking structures in biochemistry, because we have to remember sometimes they're so easy to construct that no function might be necessary for them to originate. And with that, I'm already at the end of my talk. Before I go on to the acknowledgements, I just want to come back to this idea that I think the history of the molecular components of the cell is a totally open field. I can guarantee you that almost all of the proteins that anyone here in the room is working on probably have not had their detailed history worked out, and many interesting questions and mysteries are hiding in those histories. So for me, that's a very exciting point in time to be working in this field, and hopefully it'll keep us busy for the next few years. So this is just an acknowledgement slide of my lab, and I just want to highlight the two graduate students who did most of the work I talked about today. One is Luca Schulz, who did the Rubisco work, and Franziska Zenka did the fractal work. Both of them are now postdocs. And with that, I'm ready, and I think we might even have some time for questions.